This video is for period 3, living environment. So this week we're going to get into translation. So just to review transcription, it's basically where DNA unzips, a messenger RNA molecule comes in, the bases of the messenger RNA molecule will pair up complementary to the DNA molecule, because DNA will act as a template, and messenger RNA will leave. Now we're going to move on, once it's been transcribed, into translation. And so these are the PowerPoint notes that you have that I gave you a while ago. So I'm just going through some of them today. Messenger RNA is going to leave the nucleus. This is the picture of what it would look like. And remember that in this case, there would be no T, there would be U. So we have AUG, and we have UUC and AAA. Those are what we call codons. They're read 3 at a time, and they will hook up to this molecule that's in purple, which is the ribosome. So this is the part of translation. So since we've already transcribed it, now we're going to translate it. And you could think of like Spanish class or French class, where we have to take our language and translate it into the foreign language. So this is the process by which the information coded for in the messenger RNA is used to assemble a particular amino acid sequence. What we're really making in the end here is a protein. That is the goal for all of this. It's called protein synthesis, so that's what we're making. Translation is only going to occur in the ribosome. Messenger RNA will hook up and attach to that ribosome, and the codons, which are going to be 3 nitrogen bases, will be read at 3 at a time. So you could have AUG, you could have CCG, you could have GCG. It doesn't matter, whatever the order is. But remember that messenger RNA does not have thymine. Transfer RNA is a new molecule that we mentioned last week. And transfer RNA is where basically it sort of flies in. On one end of it is going to have something called an anticodon, and on the other end of it, it will have an amino acid. And I will sort of draw on the whiteboard a visual and put another video up this week, so that'll help you, because transfer RNA can be a little confusing. Basically, it's the molecule that brings the amino acid to the ribosome, and we start adding more than one amino acid, and we create a chain that's going to develop into our protein. Transfer RNA has specific structure. It allows it to be that interpretator, and it carries with it on one side what we call anticodons. These are 3 nucleotide sequences, or 3 bases, that's complementary to the codons on the messenger RNA molecule. The anticodon of the transfer RNA align with the codons on the messenger RNA. Amino acids carried by the transfer RNA bond together in a sequence determined by the messenger RNA information. So messenger RNA is truly what's important. It's been transcribed, and you need that to basically make a sequence of amino acids. We're going to result in a chain of amino acids that are going to form a polypeptide. The cell translates the base sequence of that polypeptide protein, and that should say base. There was a typo in this online PowerPoint. Okay, if we continue on, we can sort of see a picture where we have messenger RNA and transfer RNA. You can see that messenger RNA is the bottom strand, and transfer RNA is sort of this molecule that flies in. It's sort of that orange molecule. It has an anticodon that is going to be complementary to the messenger RNA strand. So wherever you see A, there would be U, and then where you see U, it's A, and then when you see G on messenger RNA, transfer RNA will have the C. And if you look at the other end at the top, it says MET. That is basically the name of the amino acid, and we usually just refer to it as the first three letters. We don't really say the actual name of it. We just usually say the first three letters of that amino acid. If we go on, this is just another picture where it's showing that these amino acids are carried to the ribosome according to that triplet code. So you have MET that is going to be made. Transfer RNA is on the bottom. It looks sort of like a T. It's sort of a weird structure, and you can see in number 2 that it's sort of flying in and attaches with the codon on messenger RNA. So transfer RNA brings the amino acid to the ribosome so that we can now make more than one amino acid and we can make our protein, because that is our end sort of goal. Okay, I think, oh no, there was one more screen. You're going to see that we're making a chain of proteins. Those are like the sort of lighter blue squiggly lines sort of at the bottom. So it's a completed polypeptide is going to be released. We basically are going to carry this on as proteins are made all of the time in our ribosomes. So transcription is first, translation is second. Okay, we'll talk about this more, and if you have any questions, you can let me know on Wednesday.